muscle fiber will get shorter. When all muscle fibers within the fascicle will get shorter, the fascicle will get shorter. When all fascicles within the whole muscle will get shorter, that will cause the contraction of the muscle, right? The muscle will get shorter. Make sense? So that's the mechanism of muscle contraction. Okay, very important. Now tell me, if calcium is not released, troponins will not move, tropomyosin will not move, right? Myosin head will not get attached to the actin balls, right? So muscle contraction will not occur. Is it clear? So calcium is essential for what? Muscle contraction, right? And in hypocalcemia, even someone's body has calcium but less amount of calcium, that can cause paralysis of the muscle, lack of muscle contraction. Okay? So calcium is very important for muscle contraction. Now you know, right? Without calcium, muscle contraction will not occur. Okay? That can cause respiratory paralysis. Respiratory muscles will not work. You will have problem breathing. So very important. Here we have talked about this. So you see, this is a thin filament formed by two chains of actin balls, and this is that rope-like structure for tropomyosin, and these are troponins sitting on the tropomyosis. Okay? So calcium will come and will get attached to the troponins, and that will move the rope to the side. This is a thick filament, and this is a myosin tail and head part. Okay? And you see the binding sites. Now, I told you that calcium is released. Now the question is, from where the calcium is released? Okay? Each myofibril inside the muscle fiber, each myofibril is covered by tube-like structures, like network of tubes. This is the myofibril, right? And myofibril is covered by tubes. And these tube-like structures look like endoplasmic reticulum. How many of you remember endoplasmic reticulum? Remember that inside the cell? You have rough ear and a smooth ear, remember that? Around the nucleus, you have tube-like structures, right? Endoplasmic reticulum. Similar structure around the myofibril. It's like many tubes are attached to each other, network of tube, that is called sarcoplasmic reticulum. Not endoplasmic, sarcoplasmic reticulum, or SR. And these tubes are filled with calcium. So they store, sarcoplasmic reticulum stores calcium around the myofibril, because we know that calcium must be released into the myofibril. Okay? So calcium ions. And you have thick and thin filaments here. You remember that, right? Okay. So from the sarcoplasmic reticulum, calcium must be taken into the sarcomere. So who takes the calcium from here and releases into the sarcomere or myofibril? This tube-like structure. Make sense? This is the tube that takes the calcium from here to here. And this is called T-tubule. T-tubule. Transverse tubules or T-tubules. Like this. Okay. So what happens when we decide that we will contract the muscle, when we decide that I will contract this muscle, okay? I can do that, right? That's voluntary. So I send signal from my brain to the muscle, right? Calcium is released into the myofibril or into the sarcomere. From SR, the calcium will enter into the T-tubule and will be released into the sarcomere. Is it clear? Makes sense, right? So from the brain, signal goes to SR, right? And tell the SR to release the calcium into the myofibrils, right? And then calcium will do what? Bind with the antibody. Those tiny troponins, right? Will move to the side. Okay. So first thing is what? You have to decide, right? I will contract that muscle, right? So from the brain, electrical signal will go to the muscle, right? And that will tell the SR to release the calcium. Okay? Okay. So here you see, this is one myofibril, this is another myofibril, and these dots you see are thin and thick filaments inside the myofibril. Anyway, so around each myofibril you have the tube-like structures called sarcoplasmic reticulum that stores what? Calcium. Make sense? Again, you see here, this is a sarcomere in relaxed condition, and this is a sarcomere after contraction. So we know that the sarcomere gets shorter. Okay. Now, if you see under a regular microscope, not very powerful, a myofibril, you will see dark and light areas like this. You won't see everything, all those myosin heads acting. To see them, you need very powerful electron microscope, but in regular microscope, you will be able to see like dark and light areas. Okay? Why you see those dark and light bands? You all know, this is simple. When you see a tissue under the microscope, right, you pass what from the bottom? Light. Right? You remember, the light goes like this. So this is the tissue here. You pass the light from the bottom, right, and look from the top like this. So when light passes through the slide, where you have more tissue, more or less light will be able to pass. Less. When you have less tissue, more light will be able to pass, right? So where less light passes, that area will look darker. Make sense? Where more light passes, will look brighter, lighter. So that's why you see these dark and light bands. Make sense? So you see here, this area is darker than this area. This area is light. Why is that? If you see the structures here, you have thick and thin, both filaments in this part, right? You have both. So more tissue is here. Make sense? Less light will pass. And let's see here. Here you only have thin. No thick filament is present in this part. So that's why this area looks lighter. Make sense? So where you have only thin, looks light. Where you have both thick and thin, will look darker. Make sense? That's why we see where you have Z discs, that area will look dark. So you can see the Z discs because they are like thick dark lines. Less light passes through it. That's why we see those bands, dark and light bands, and we name them different, bands like Z, H, A or Z. You don't have to remember. You know the reason, right? Why? Some areas are dark, some areas are light. Okay? Just know that. Okay, now we'll talk about a very important structure that is called a neuromuscular junction, NMJ. Its name is telling you what is it. This is a neuron. Anybody remember a neuron? Okay, good. So this is the cell body, nucleus, right? And the long process is called what? Axon, right? Remember axon? And short branching processes are called what? Dendrites. So these are the dendrites, okay? So this is a neuron. This is the axon terminal. Terminal. These are axon terminals. Make sense? Okay. And axon terminals end on the muscle fibers. So these are the muscle fibers. Muscle fiber. Make sense? So tell me, how many axon terminals this neuron has? Three, right? So how many muscle fibers are getting signal from this neuron? Three. Make sense? This is a small neuron. There are some neurons, bigger neurons, they have like 1,000 axon terminals. Make sense? So from one neuron, you have 1,000. So those neurons have more terminals, they will innervate more muscle fibers. Make sense? They will give signal to more muscle fibers. There are some small neurons, they have only few axon terminals, like this one. Okay? So only few muscle fibers will get signal from that neuron. Is it clear? So the number varies a lot. One neuron and the muscle fibers innervated by it or getting signal from it together is called a motor unit. So this is one motor unit. Make sense? One neuron and the muscle fibers getting signal from it. Is it clear? So this motor unit consists of one motor neuron and how many muscle fibers? Three muscle fibers, right? That is one motor unit. And the area where the axon terminal talks to the muscle fiber, that is called a neuromuscular junction. So this is a neuromuscular junction. This is another neuromuscular junction. So these are called neuromuscular junction. Is it clear? Neuron is talking to the muscle. You tell me from this drawing, the axon terminal is directly attached to the muscle fiber or there is a gap? There is a gap, right? Remember, there is a gap. That is called synaptic cleft. We'll talk about that. Just know that the axon terminals are not directly attached to the muscle fiber. There is a tiny gap. That's why I did draw this way. Okay? Now, what I will do, this is very important. I may ask you to draw this picture in your exam. So I'm just going to look at this part, neuromuscular junction. Okay? So first I'll draw this part, then I'll draw this part. Let me use the same color, dark. So this is the axon terminal. only this part, axon terminal. And this is the muscle fiber. Muscle fiber. This is the gap. This gap is called what?
Synaptic gap.
Synaptic what?
Synaptic gap.
Gap or
Cleft.
cleft. You know cleft? Have you heard that? Have you heard cleft palate? Right? There is a gap, right? Or cleft lip. Gap. Also called synaptic gap. More often we use the term cleft. Okay. Now, at the axon terminal, you have vesicles, round membranous sacs. You know vesicles. Vesicles. We talked about it when we talked about the cell. And these vesicles are filled with a neurotransmitter, neurochemical, very important, that is called ACH. Do you know what is this? Acetylcholine. Have you heard this? Acetylcholine? It's a neurotransmitter inside the vesicles at the axon terminal. And on the muscle fiber, you have the receptors. These are called ACH or acetylcholine receptors. On the muscle fiber, you have the receptors. Make sense? Okay. When we send signal that will cause the muscle contraction, electrical signal comes from the brain through the nerve, neuron, like this. Look at this. Electrical signal will go like this. Will go back. Will pass through the presynaptic terminal and will our axon terminal and will go back like this. Electrical signal will go like this. So when the electrical signal goes like this, passes through this terminal, what happens? Calcium channels get opened and calcium from the synaptic cleft will enter into the terminal. Calcium channels will get opened and calcium ions will get in. This calcium is different calcium than the calcium we talked about actin myosin in muscle contraction. This is different. Now, why calcium should enter? Because that calcium will cause the rupture of these vesicles, breakdown, exocytosis. So, ACH molecules will be released into the cleft. So these are the ACH molecules come out from the vesicles. So to rupture the vesicles and release the calcium, you need, release the ACH molecules, you need calcium. That's the function of calcium. Okay, now you see what happens. ACH molecules are released here and these are ACH receptors. So what will happen? The ACH molecules will bind with these receptors. Because these are ACH receptors, they are waiting for the ACH molecules. Their shape is similar to the molecules. Okay, so the binding will be like key lock. We talked about that before. When that binding will occur, ACH receptor molecule binding will occur on postsynaptic membrane. Sodium channels will get open and sodium will get in. Plenty of sodium will get in. Sodium from the cleft will get into the muscle fiber. Okay, let's quickly review. Okay, so this is what? A neuron, right? And this is a muscle fiber. One neuron and the muscle fibers getting signal from it together is called what? A motor unit. Remember? Motor unit. So this is one motor unit. Some motor units are very big, right? Like 1,000 muscle fibers are getting signal from one neuron. Okay. And this part of the neuron is called what? Axon terminal. Make sense? And axon terminal talks to the muscle fiber, right? And there is a gap. That gap is called what? Synaptic cleft. Right? Here. And this end, axon terminal, is also called presynaptic terminal. Let me write it down. Axon terminal or presynaptic terminal. So presynaptic or axon terminal. Same thing. Because before the synapse. Okay. Now you tell me. Axon terminal has vesicles, right? Filled with what? ACH. Acetylcholine vesicles, right? And on postsynaptic membrane or on the muscle fiber, this is called postsynaptic membrane. This membrane, postsynaptic membrane. So presynaptic terminal and this is presynaptic terminal or axon terminal. This is postsynaptic membrane, the muscle fiber. On postsynaptic membrane, you have receptors, right? Called what? ACH receptors. Make sense? Because they are for ACH, not for other molecules. Now, when electrical signal is sent from the brain, that travels through the neuron and goes through the axon terminal like this, and that will open which channels here in presynaptic membrane? Calcium. Calcium will get in. Make sense? To do what? Rupture the vesicles. Is it clear? Exocytosis, right? That will release the ACH into the cleft. And ACH will bind with the postsynaptic receptors. That will open what? This is important, you see. On postsynaptic membrane, the sodium channels will get open. Okay? Sodium will get into the muscle fiber. Did you get it? Sodium will enter into the muscle fiber. This is very important. Why? Because look at this. I am drawing a muscle fiber again. This is the muscle fiber. Okay. Now sodium got in. Okay. From outside. Sodium influx. Sodium is positive or negative? Positive, right? Now you tell me a simple question. If sodium gets into the muscle fiber, inside the muscle fiber, it will become more positive or less? More, because sodium is getting in, right? Positive is getting in. So inside the muscle fiber will be more positive. Is it clear? Okay, so let's see here. If I draw a curve to show, this is called resting membrane potential, RMP, before sodium got in. Okay? This is the condition inside the muscle fiber before sodium got in. Usually the resting membrane potential is minus 70 millivolt inside the muscle fiber. Before, sodium channels got opened. Now sodium got in. So when sodium is getting in, if I draw positive this way, negative this way. When sodium is getting in and I am looking inside the muscle fiber, so which way it will move? Inside the muscle fiber will move towards positive. Make sense? Sodium is getting in. So inside the muscle fiber is getting more positive. Are you positive? Got it? Because sodium is positive, right? So getting in and I am looking inside, nothing else. So inside is moving towards positive, right? So this curve will move this way. That is called depolarization. Have you heard this? Very important phenomena, depolarization. Moving towards positive. And after that, what will happen? The sodium channels will get closed. So no more sodium channel is open. But inside is now more positive. So depolarization, it is here now. Okay? Then sodium channels will get closed, no more sodium will get in. Then next, what will happen after sodium channels got closed? Potassium channels will get opened. Okay? And now this is opposite. You see, potassium will get out. Potassium will get out. Now you tell me, potassium is positive or negative?
Positive.
Look at that. Potassium is what?
Positive.
Also positive, right? You see K plus. Potassium is also what? Positive. So when positive potassium gets out, inside the fiber, muscle fiber will be what? Will move towards negative. Make sense? Less positive. So this will go which way? Up or down? Down. Make sense? And this part is called repolarization. Moving towards negative, that means resting membrane potential. Moving towards the resting membrane potential. Okay, let's now quickly go over. So when ACH molecules will bind with the receptors, which channels will get opened here? Which channels getting open? Sodium, right? And sodium is getting in. That is same thing I showed you here, right? First, sodium will get in. When sodium will get in, what will happen? Depolarization. Is it clear? Then, when the depolarization will reach here, sodium channels will get what?
Negative.
Closed. Make sense? But which channels will get open now? Potassium. Make sense? And potassium will get out from the muscle fiber. Is it clear? So this will move downwards, right? Because positive is leaving now. When positive is getting in, depolarization. When positive potassium is leaving, repolarization. Make sense? So influx of sodium causes what? Depolarization. Efflux or outflux of potassium will cause what?
Repolarization.
Depolarization. Is it clear? So this is due to influx of sodium. This is due to efflux of potassium or outflux of potassium. Repolarization. And these two things together is called the action potential. So action potential has a depolarization and a repolarization. This whole part is called what? Action potential. This is very important in biology. particularly in electrophysiology. Depolarization, repolarization, very important. Okay, if you don't understand, it's a problem. Now, you can ask me why we need inside the muscle fiber, why we need depolarization and repolarization inside the muscle fiber. Why we need it? You remember I said that when we decide that we will contract the muscle, we send signal from the brain to the muscle, right? And that causes the release of calcium. Remember, we'll bind with troponin. Tropomyosins will move to the side. And who releases the calcium? Who stores the calcium? SR, sarcoplasmic reticulum, those tubes. Remember that? So, signal goes there. And calcium is released into the muscle fiber, and that causes the muscle contraction. Now, depolarization is the signal that will tell the SR to release the calcium. So, just know that without depolarization, calcium will not be released